All right, thanks, Brian. So it's my pleasure to present to you today some results from a project to describe the ciscoes of the Laurentian Great Lakes and Lake Nipigon. For the purpose of this talk, I'm not going to focus much on Lake Nipigon. I'll focus on each of the individual Great Lakes. So much of what we know about the diversity in terms of the taxonomy and systematics of Coregonid ciscoes comes from the work of Walter Keltz. Published in 1929 was this famous monograph based on some of his doctoral dissertational work that was completed in the 20s. So he collected roughly 9,000 specimens between 1917 and 1924. He measured painstakingly 32 morphometric and meristic variables, and I'll describe to you in a subsequent slide what I mean by that. 9 species were described. They were given proper scientific, genus, and specific names, 4 of which he named himself, and 12 subspecies were described, and I'll explain what I mean by that as well. So this work was seminal. It remains relevant today. It's a hugely important and valuable resource, but it's complicated to employ if any of you have tried to use it. It's also outdated. As I mentioned, Keltz collected his samples in the early 1900s, so almost a century ago. There have also been several extirpations, local extirpations and extinctions, and there have been changes in taxonomy in the Great Lakes. There also have been changes in morphology within forms over time and among the lakes. A couple of caveats to Keltz is that there was no statistical treatment of the data, so much of his data is presented in the appendices as frequency distributions. In addition, only a single key was presented for all of the forms in all of the lakes, and if any of you are familiar with ciscoes, you realize that's problematic, and I'll show you throughout this talk why that is and how we've taken a slightly different approach. So the objectives today are first to provide you with an updated status of the ciscoes in each lake, secondly to identify notable changes in morphology, and third, to present to you some management recommendations and research recommendations as managers become more engaged and involved in Coregonid cisco restoration in the Great Lakes. So just want to set a bit of the stage here, talk about the relevance to reestablishment of this project. If we think about restoration in terms of a scale, I'll present to you the two ends of the strategy in terms of restoration. The first end of that spectrum is to stock locally adapted forms into suitable habitats. So an example would be the deepwater bloater being stocked into suitable deepwater habitat in Lake Ontario. The other end of that spectrum is to stock a less differentiated, more genetically diverse form into the open lake and sort of mimic the early stages of evolutionary divergence. So these are kind of the two bookends on that spectrum of strategies. And the strategy will differ depending on whether we're trying to reintroduce shallow water ciscoes, like Coregonid sartidi, for example, versus the deep water forms. So just a few basic concepts before I get into some of our results. Keltz named, whether it's fortunate or unfortunately, these different forms as species, giving them a genus name, the genus Coregonus, and specific names. So shown on the right here are the five of the common forms. We're referring to them rather than species as forms, and we're using the specific part of that formal name only in the colloquial sense to refer to each of these different forms for convenience. So RTD is, rather than Coregonus RTD, we're referring to it as RTD as a form here, as well as minor forms rather than subspecies. So things like the albus or the deep-bodied form of RTD, we would refer to it as a minor form here. Second thing to point out is that historically, Keltz considered the Great Lakes Basin as a whole as containing a cisco flock, so a flock of different forms of ciscoes. Here we recognize variation within each of the individual lakes, and so refer to flocks within lakes. And so that's a difference between how we're treating these animals versus how Keltz treated them. So we have 3 different sets of data that we drew on for this project. I mentioned here morphometric and meristic data. So what I mean by morphometrics are linear measurements. So things you can measure like fin lengths, eye diameters. Meuristics refers to count data, so things like gill raker number, counts of gill rakers. So we have data from 3 different time periods. As I mentioned, Keltz's data from the 1920s, roughly 9,000 individual fish. We have data from the 1950s to the 70s collected by Stanford Smith, about 1,400 individuals. And we have data from the 2000s, so the contemporary data set, roughly 3,300 individuals. So in total, we have about 13,000 individual fish represented in this guide to varying degrees. The one thing I want to point out here is that morphometric and meuristic data are important. They're not just markers, but they're important because several of the characteristics or traits we're measuring here are adapted to things like trophic ecology, so what these animals are feeding on. Their head morphology, their jaw morphology adapts to the kinds of things they're feeding on. Also, some of these measurements are adapted to the kinds of environments these animals are living in. So they're very specifically selected for and adaptive, and so that's why they become important to managers as they consider source populations for reintroduction into particular habitats, for example. Okay, so the guide is organized around 8 morphometric and 1 meuristic variable, the meuristic variable being gill raker number, that are common to all 3 of those data sets. So there's a huge amount of effort that went into digitizing historic data, Keltz's data, data that was on IBM punch cards, and then summarizing those data into statistics, and many of them had to be corrected for so that they were comparable between Keltz's data set and our data set. For example, we found out that Keltz used dividers to measure some of the variables, some of those linear measurements, whereas we used a vernier caliper, and so there are slight differences in some of these proportions. So we had to develop means of correcting those. So we ended up with 9 variables that are common to all 3 of those data sets. We described 8 major and 2 minor forms, the 2 minor forms being the albus form and the manitolinus form of Artidi. Our guide is organized by lake, so we have individual lake sections, and I'll show you what that looks like. It's well illustrated thanks to our co-author Paul Vecce, and I'll show you some examples. We are able to describe morphological changes from the 1920s to the 2000s. And we provide a fairly detailed review of the taxonomy, the distribution, and the ecology of these animals in each of the lakes. So this is what some of the sections look like. We provide color illustrations. These are scientifically accurate illustrations. You can actually do the morphometrics on these illustrations and get appropriate proportions for each of the individual variables. We also produced a set of distribution maps. So shown in red is the historical on your left-hand side, the historical distribution, and on the right, the contemporary distribution. The historical data is based on anything published or available up until the 2000s. The contemporary distribution is based on USGS trawl data that's been unpublished, some of it has been published, as well as anything that was published post-2000. So we produced that for each of the main forms. And then within lake sections, that's where I'm going to focus on the bulk of the talk here and describe the cisco flock within each of those lakes. So this is an example of the forms that are extant in Lake Superior. So we've got these kinds of illustrations, not for every form, but for the majority of them. So the next few slides, I'll focus on some results. We tried to pick out some results that are relevant to managers as they begin to think more about restoration of coregonate. So there's a couple things I want to highlight here. The first column, the left-hand side of the screen, represents the major form, and shown across the top are the lakes. The blue boxes here represent the extant forms in each of the lakes. 2 things to notice is first that Superior and Lake Nipigon have almost the complete set of historical forms extant. This yellow boxes here show the extinct and extirpated forms. Lakes Michigan and Ontario were particularly affected. The green boxes here show the uncertain status of Nigropinis and Regardae in Lake Superior, and I'll circle back and touch on that when I talk about Lake Superior specific results. In Lake Huron, the data suggest, and this is building off a suggestion by Tom Todd in the 1980s, that the deep water forms in Lake Huron are introgressed into a hybrid swarm, and I'll present to you some results there. Most of the data are summarized within the guide to support that conclusion. So over the next few slides, I'll talk about some of the specific results for each lake. So Lake Superior, we see no extirpations. However, contemporary Artidae had 3 fewer gill rakers than Artidae collected by Stanford Smith in the 50s and Walter Kelts in the 20s. So the change in Artidae has occurred since the 1950s. What that means from a biological standpoint, we don't know, but gill rakers tend to be related to feeding. And so there could be some implications there with respect to the ecology of this animal. I mentioned too that the Niagara-Pinus and Regardae forms, the status is unknown in Lake Superior, in part because as USGS went through some of their database, they found several individuals that represent those historic forms that were previously thought to occur in the lake. So perhaps some more surveys can help shed light on the existence of those 2 forms. Albus and Manitolinus, those are the 2 forms that I mentioned of Artidae. Some recent work by Dan Ewell suggests that those forms may indeed occur in Lake Superior. With respect to Lake Michigan, we've seen 6 forms extirpated over the last 100 years, so pretty big change. Artidae and Hoyai remain extant in the lake. With respect to Artidae, 5 of the 8 metrics that we used for contemporary Artidae diverge notably from those collected by Walter Kelts. So some pretty big changes in the morphology and presumably the ecology of these animals. And 3 other metrics diverged marginally. For Hoyai, we see only 1 change, and that's in pelvic fins becoming shortened. And interestingly, all of the extant Hoyai populations showed this same change in pelvic fins becoming shorter since the 1920s. Not sure, again, what that means from a biological perspective, but there's some common selection pressure that may have acted on that variable. Also, there's some evidence of temporal change in gill raker number over time. Some of you may recall some of the work in the 1980s showing these changes in gill raker number, declines in gill raker number. The contemporary data show that they tend to be more in line with what they were during the 1950s in recent times. For Lake Huron, the Artidae form is extant, but we recognize that it may indeed not be the typical offshore pelagic spawning Artidae. A lot of the data that we got are for these near-shore albus-like, so these deeper body shoal spawning forms of Artidae. The status of typical Artidae is unknown. 4 of 8 body metrics for contemporary Artidae diverge notably from those collected by Kelts in the 20s. The deepwater forms, as I mentioned, seem to have introgressed, and it appears to have happened sometimes after the 1950s into a hybrid swarm. The hybrid tends to be more Hoyai-like in its morphology, so more like bloater, but it tends to be intermediate among all of those deepwater ciscoes that were extant during the 1950s. So some considerable changes in Lake Huron. For Lake Erie, we've seen no evidence of remnant albus or typical Artidae. So some of you may be aware, a few of you have participated in some of the work we've done here on Lake Erie, but only 29 ciscoes have been collected since 1995. We had the opportunity to look at about 20 of those that were collected between 2003 and 2015, I think. And they tend to be morphologically similar to that hybridized form from Lake Huron. So it's likely that these animals may have moved downstream in the system. So based on the data that we have summarized and compared back to Kelts, it's unlikely that there are remnant Artidae in Lake Erie. Lake Ontario, we saw 3 extirpations. Typical Artidae still are extant and remarkably stable morphologically. Several of the variables we measured are identical, including the standard deviation to those measured by Kelts in the 1920s. And we sampled some of the same populations that he sampled. So extremely stable, despite the fact that the other forms and the deepwater forms have changed. Relatively few Hoyai have been reintroduced, and I say relatively few. There have been tens of thousands of animals introduced to the deep water, but in comparison to the historical population, it's probably very few. So just to summarize those changes, we see 21 instances of notable change, and again, there I mean by that 2 standard deviation differences from the mean collected by Kelts in the 1920s. If you convert that to a rate of change per form, it's roughly equivalent across the extant forms of ciscoes, so between 0.2 and 0.25 changes per form. Some forms have been remarkably stable, like the Lake Ontario Artidia that I showed you. Also, bloater generally, besides the pelvic fin change, have been remarkably stable. Artidia accounted for nearly half of those notable changes, but there's a caveat here in that some of the samples that we collected during contemporary time, or some of the populations we sampled during contemporary time, are not the same ones sampled by Celts. His sampling focused on offshore pelagic forms, and we have mixed in with that a lot of sampling on the near shore, more albus-like forms, and so we need to do a little bit more work there. In fact, some of those Artidia populations may indeed have been more stable than our analysis suggests. So I'm just going to wrap up the last couple minutes here with some basin-wide management needs. The first is a question: do we need a coordinator given the efforts going on with respect to reestablishing gorgonids in the deep water? We have said that we would help train field biologists, so some of these morphometrics. We're not recommending that morphometrics be done through all the assessments. What we are recommending is that they're pretty much the only tool we have right now for really differentiating among these different forms, and they can be helpful if we're trying to identify source populations or assess animals that have been stocked. We need to develop management-based assessment priorities in each of the lakes, and and I'm helping John organize a workshop to do just that this fall. So some of the questions we may want to tackle is albus form extant in Lake Huron? What is the status of typical Artidia in Michigan and Huron? We also need to understand the data gap for juveniles. All the data I presented to you, even from Celts and our work, is for adult-sized individuals, so greater than 200 millimeters. We really can't yet differentiate well the juveniles. We need to fill in some data gaps in existing collections. There's a huge pile of animals at the University of Michigan Museum of Zoology that were collected during the 50s by Stanford Smith. They don't have accession numbers, they're not accessible, and they'll help fill in some of these gaps. We need to identify stock structure for all the forms and the behavioral basis for that structure. And then finally, assess some of the plasticity in Artides, essentially redoing some of the breeding experiments that Tom Todd did during the 1980s. So these are just a few of the management relevant results that came out of this project. And then finally, we expect to have this wrapped up and published during the summer of 2016. There's been a lot of people, especially Randy Eschner, who have put a ton of work into getting this to where it is now. So thank you, and with that I think we have a minute or so for questions, if anybody has any.